Welcome to the deep dive. Today we're tackling something really fundamental, WNT signaling. It's this pathway that's just absolutely critical for, how our bodies develop, how tissues stay healthy, and how they repair themselves.
Absolutely. But the big hurdle has always been that WNT proteins themselves are, well, incredibly difficult to work with.
Difficult how?
Well, they're sticky, they're tough to purify, they don't always hit the exact receptors you want them to target. And then there are these essential fat modifications that add another layer of complexity.
Right. So that's really hampered research and, I guess, therapeutic development too.
For sure. It's held the field back quite a bit. People have tried making WNT mimics before, but those earlier versions often had their own limitations.
Okay, so that really sets the stage for some pretty exciting new research we're looking at today. We've got a few sources here, a correspondence piece in Cell Chemical Biology, a research article in Nature Communications, and a review from iScience. And they all describe, a totally new generation of tools.
That's right. We're talking about what they call a complete set of WNT mimicking molecules. These can apparently activate every single frizzled receptor involved in the main WNT pathway.
A complete set of keys, essentially, to unlock all those different WNT doors.
Exactly. And not only that, they've also built this novel platform that combines WNT mimics with mimics of another key player, RSPO, all into one, like, supermolecule.
Wow. Okay. So our mission for this deep dive then is to really get under the hood of these next-gen tools, understand how they were made, and importantly, what they might mean for research and maybe even new therapies down the line.
Precisely. These papers give us a really detailed look at the engineering and the potential applications.
All right, let's dig in. First up, developing this comprehensive set of frizzled-specific WNT mimetics. The idea was to hit all 8 of those key beta-catenin activating frizzled receptors, right? FCD1, 2, 4, 5, 7, 8, 9, 10. How did they actually build them?
They used antibody engineering.
Yeah.
Antibodies have these arms that grab specific targets,
Yeah.
right? So they played around with different formats, different ways of putting antibody fragments together.
Different formats. Like what?
Things like tandem SCFE-FC, which is sort of a smaller engineered bit linked to a standard antibody part, FE-IgG, Fab-IgG, FE-Fab. Uh, each one has a slightly different shape.
And why different shapes?
Well, the geometry matters. It affects the distance and the angle between the arms that bind to the frizzled receptor and its co-receptor, LRP. They needed to find the format that Beth brought those two together to kickstart the signal.
Okay, makes sense. And did one format work better than others?
Oh, definitely. The FEIGG format was the clear winner. It showed the highest activity, especially when they combined an antibody part that binds LRP, let's call it L, with one called F12578 that binds to a bunch of frizzles, FZD1, 2, 5, 7, and 8.
So a broad spectrum frizzle binder combined with the LRP binder in that specific FEIGG structure.
Exactly. And the difference was pretty stark. Just to give you numbers, the FEIGG version of this LF12578 had an EC50, that's a measure of potency, of about 0.033 nanomolar.
Okay.
Compare that to the SCFEFD version, which was 0.70 nanomolar. So the FEIGG was, much, much more potent.
Wow, that's a huge jump. That means you need a lot less of it for the same effect, I imagine.
Precisely, which is a really big deal when you start thinking about potential drugs. Less drug needed is usually better.
Okay. So they picked this winning FEIGG format, and then they basically made a whole toolkit using it.
That's the idea. They took that LRP binder, L, and paired it with different FZD binding antibodies.
Mm.
So there was F127 for FZD1, 2, and 7, F58 for FZD5 and 8, F4 for FZD4, F49 for FZD4, 9, and F10 for FZD10.
Covering all the bases.
Right, giving them a specific tool for basically the whole family.
Right.
And what was quite neat was that some of these, like the ones for FZD4, 9, and 10, really only worked well when they tested them in cells that were engineered to make lots of those specific receptors.
Ah, so that confirmed they were actually specific, hitting their intended targets.
Exactly. It really highlighted the precision they'd achieved.
Okay, impressive toolbox.
Hmm.
So where did they take it next? The first results you mentioned were about effects on salivary glands. Why start there? Kind of specific.
It was actually a bit serendipitous, it seems. When they first tested some of the broader mimetics like LF12578 and LF127 in mice, they noticed something odd.
Odd how?
The mice started grooming themselves a lot more around their mouths, getting damp fur. It made the researchers think, maybe this is affecting saliva production.
Interesting observation. And did it lead somewhere?
It really did. They followed up with proper experiments and found that giving these WNT mimetics actually caused a statistically significant increase in salivary gland weight. It's called hypertrophy. the glands got bigger.
Bigger glands. Did they look different inside?
Yeah, when they looked at the tissue microscopically, the acinar cells, those are the saliva-producing cells, looked slightly enlarged. They had more prominent nuclei and what's called more basophilic cytoplasm.
Which suggests.
It suggests the cells were potentially more active, maybe making more proteins, the components of saliva.
Okay, so bigger glands, maybe making more saliva. Was it just the cells getting bigger, or were there more cells too? Was there increased cell division?
Good question. They looked at that. They saw a temporary spike in dividing epithelial cells around day 7 after starting the treatment with LF12578, but it wasn't sustained later on.
So an initial burst of growth, maybe?
Seems like it, yeah,
Yeah.
contributing to the overall size increase. And to investigate further, they used salivary gland organoids.
Ah, the mini organs in a dish.
Exactly. These 3D cultures mimic the real gland, and they found that LF12578 and especially LF127 really boosted the growth and expansion of these organoids, and it depended on the dose.
And did that connect back to the specific frizzled receptors?
It did, nicely. Those salivary gland organoids happen to express high levels of SED7, SED1, and SED2, the very receptors targeted by LF127 and LF12578. So it strengthened the link.
Very cool. And you mentioned therapeutic potential, Sjogren's syndrome.
Right. That's an autoimmune disease where the immune system attacks glands like salivary glands, causing severe dry mouth. They tested LF12578 in a mouse model of Sjogren's,
Yeah.
and it significantly increased salivary gland weight in those diseased mice too. So it hints at a possible new way to help treat that condition, which is pretty debilitating.
That's really promising. Okay, let's shift to the next big development. These superagonist WNT molecules. Sounds powerful. What makes them super?
Yeah, this was a clever idea. The researchers knew that WNT signaling often collaborates with another pathway, the R-spondin, or RSPO pathway.
Right, RSPO boosts WNT signaling, doesn't it?
It does. RSPOs basically help clear inhibitory receptors off the cell surface, making the cell more sensitive to WNT signals. So the idea was, what if we combine the WNT activation and the RSPO effect into a single molecule?
One molecule doing both jobs. How'd they build that?
They engineered fusion proteins. They essentially linked their SED-binding antibodies, like that F12578 we talked about, to a modified piece of the RSPO2 protein called RSPO2RA. Now, here's where it got really interesting, and actually quite unexpected.
Oh.
They initially thought maybe this fusion protein, F12578 RSPO2RA, might actually interfere with or block signaling because it binds frizzled. But when they tested it alongside the natural WNT protein, WNT3A,
What happened?
it enhanced the WNT3A signaling. Especially at lower doses of WNT3A, it acted like a booster.
That is surprising. So it didn't block, it helped. How?
They figured out that the F12578 RSPO2RA fusion was actually increasing the amount of FCD receptors on the cell surface. So in that context, it was behaving more like an RSPO mimetic, amplifying the WNT signal.
Huh. So leveraging the frizzled binding to actually boost the pathway.
Exactly, and this discovery led them to design the superagonist WNT molecules. The main example is LF12578 RSPO2RA. Think about it. It has the LRP binder, the FZD binder, F12578, and the RSPO mimetic function, RSPO2RA, all built into one molecule. It's trispecific and technically hexavalent because of all the binding sites.
Wow, a real multitasker molecule. What could it do?
They tested it in organoid cultures again, but this time from a whole range of tissues: mouse and human small intestine, colon, esophagus, liver, kidney, salivary gland.
And the results?
Pretty amazing. These single superagonist molecules could completely replace both WNT3A and RSPO in the culture media needed to grow these organoids.
Replace both. That's huge for organoid research.
Definitely. They even showed that if you block the cell's own internal WNT production, the superagonist could still drive robust organoid growth all by itself. And importantly, the organoids grown this way could still differentiate properly into various cell types. They weren't just growing uncontrollably.
That's incredible. Massive implications for regenerative medicine and just basic research using organoids. Okay, let's switch gears now to our final main topic, WNT mimetics for bone. We know WNT is super important for bone health, right?
Absolutely critical. WNT signaling drives the cells that build bone, the osteoblasts, and helps put the brakes on the cells that break down bone, the osteoclasts. So it's a prime target for bone diseases.
So the researchers aimed WNT mimetics specifically at bone.
Yes. They started developing bispecific WNT mimetics ones that bind both frizzled and LRP, specifically to try and increase bone mass.
And they had some early success with AAV delivery. Viral vectors.
They did. An early proof of concept used an AAV to deliver a molecule called 18R5-DKK1C. It combined an FZD binder with part of a protein that interacts with LRP. And in mice, it did increase bone mineral density and bone volume.
Any side effects with that approach?
They noted a temporary increase in liver weight, but importantly, when they looked at the liver and gut tissues, they seemed normal, no signs of abnormal cell growth. But while promising, that AAV approach led them to develop, let's say, more drug-like molecules.
Which brings us to the IgG-based bispecifics like FAL5, FBL6. What do those letters and numbers mean?
Right, so FA and FB are different frizzled binders. FA is broader, hitting FCD1, 2, 5, 7, and 8. FB is narrower, just FCD1, 2, and 7. And L5 and L6 are different LRP binders.
Okay. And they optimized the design of these two.
They did. They found that attaching small antibody fragments, called VHH domains, to the main SD-binding IgG antibody boosted activity. Specifically, attaching them to the light chain part of the antibody worked best.
And were these potent?
Very potent. Molecules like FAL6 and FBL5 strongly activated WNT signaling in lab cells, including osteoblast precursors, the cells that become bone builders, much more potent than natural WNT3A, actually.
More potent than the real thing. And importantly, did the separate parts work, like just the LRP binder?
No. The LRP binders alone, L5 or L6 on their own, were inactive. You needed both the frizzled and LRP engagement together.
Okay, highly potent specific molecules. What happened when they put them into actual animals?
Really positive results in normal mice. Treating them with FAL6 led to significant increases in bone mineral density, whole body, spine, femur, plus thicker cortical bone, which is the strong outer layer.
Stronger bones, basically.
Exactly. They also saw higher levels of a bone formation marker in the blood, and the femurs were mechanically stronger, harder to break. And interestingly, even the mimetic with the narrower FCD target, FBL5, also effectively boosted bone growth.
That's quite something. Even targeting fewer frizzleds worked well for bone. What about disease models like osteoporosis?
That's where it gets really clinically relevant. They use the overreactomized mouse model, which mimics postmenopausal osteoporosis,
Yeah.
and both FAL6 and FBL5 significantly reversed the bone loss in these mice.
Reversed it, so increased bone density?
Yes, increased whole body and femur bone density and improved the biomechanical strength of the bones, made them stronger again.
Could they combine these with existing drugs?
They tried that too. They combined the WNT mimetics with either an anti-sclerostin antibody, another type of bone-building drug, or a common bisphosphonate drug. And they saw synergy,
Meaning?
meaning the combination worked better than either drug alone. They got bigger, more sustained increases in bone density, potentially a much more powerful treatment approach.
Wow, okay, and bone repair, like fixing fractures.
Yes, they looked at a fracture model too. If they gave the WNT mimetics, FALS6 or FBL5, 2 weeks after the fracture occurred.
Why 2 weeks after?
Good point. We'll come back to that. But when given 2 weeks later, it enhanced bone healing. They saw a bigger callus, that's the initial healing tissue, more bone formation within the callus, better mineralization.
So it sped up or improved the repair process. Now, why not give it immediately?
That's the surprising part. When they gave the mimetics right after the fracture, it actually seemed to inhibit the early callus formation.
Really? Hindered healing if given too early.
Yeah, it suggests that the timing of WNT activation is really crucial in fracture healing. You might need that initial inflammatory phase before boosting the bone building phase.
Fascinating. Biology is complex. Did they look at how these mimetics were working in the bone at the gene level?
They did some gene expression analysis. FBL5 turned up genes involved in making osteoblasts, similar to anti-sclerostin. But it also showed a stronger increase in genes like axin-2, a direct WNT target, and a PSA67, a proliferation marker. Suggests maybe a slightly different or broader mechanism of action.
Okay, so incredibly promising results for bone, both building it and repairing it. Now, these WNT agonists seem to have potential way beyond bone, right? The review article touched on other areas.
Absolutely. The review paints a broader picture. There are apparently clinical candidates based on related technologies already in trials. One for liver injury, targeting RSPO signaling, and another WNT mimetic for ulcerative colitis, targeting FCD5 and 8 specifically.
Clinical trials already, okay.
Yeah, and they mentioned potential for targeting FZD4 to help stabilize leaky blood vessels, like in the retina or maybe the brain. And of course, as we discussed, targeting FCD1, 2, and 7 for salivary gland regeneration.
So a wide range of possibilities.
Definitely. The big advantage of these synthetic agonists is they give potent pathway activation, but they also have much better properties for actually being developed into drugs manufacturing, stability, delivery, compared to trying to use the natural WNT proteins.
This has been a really fantastic deep dive. So to sum up, we've seen the creation of this comprehensive toolkit of FCD-specific WNT mimetics, giving really precise control.
Check. And then the invention of these superagonist molecules combining WNT and RSPO effects, which showed amazing results in organoid cultures.
Right. And the in vivo data is compelling, stimulating salivary gland growth, and especially the really strong effects on building bone, reversing bone loss, and even enhancing fracture repair under the right conditions.
The bone data, particularly the synergy with existing drugs and the reversal of osteoporotic bone loss, is very exciting from a therapeutic standpoint.
Yeah, absolutely. For you listening, a couple of aha moments for me were definitely that surprising finding where the RSPO fusion protein actually boosted WNT signaling by getting more receptors to the surface.
That was definitely counterintuitive at first glance.
And also how effective even the narrower FCD targeting mimetics like FBL5 were for stimulating bone growth. It shows real specificity potential.
It does. And knowing that related molecules are already in clinical trials for liver and gut diseases really brings home the potential here. This
Yeah.
isn't just lab bench stuff anymore. It's moving towards potentially helping patients.
Okay, so here's a final thought to leave you with. WNT signaling is involved in so many different biological processors, right? And now we have these incredibly precise tools to turn it up, potentially in very specific ways. So thinking beyond bone and salivary glands, what other conditions, maybe ones that seem intractable now, might become treatable if we could carefully and selectively activate WNT pathways? Something to ponder,
A lot of possibilities out there.
definitely. Thanks for joining us for the Deep Dive.